We in there. All right. Don't y'all be telling nobody about my little rig now. All right. So real quick, I want you to turn to page in your notebooks. I need you to turn to page 88. Okay. Turn to page 88 for me in your notebook. Turn to page 88. We're gonna talk about this project first. On page 88, we're going to talk about this project first.
Still on unit 1 or whatever?
This is we're in unit 3.
Unit 3,
Yeah, we're in unit 3.
Page 1 again?
Page 88. Page 88. Page 88. Alright, so on page 88, this is your written instructions for your biomolecules project. Remember, that is a major grade. A day will be due on October 15th. B day is due October 16th. Okay? It's a major grade. The 16th is the same day you'll take your test on this unit. Now, all you need is a poster board, a regular poster board, not a trifold, but a regular poster, and then you have to fold it the way you see on the bottom. It says, and it gives written instructions, and then there's also a picture there. I think I'm gonna make a video to actually, so you guys can actually see me fold it with some regular paper, so you can try to follow along with your poster board.
How big the poster is?
Just a regular poster board. Just a regular poster.
Just one?
One, just one. All right, now once you fold it, it'll look like it has 4 rooms, and it'll be 4 different rooms. Each one of those represents one macromolecule. So you have one for carbs, one for lipids, one for proteins, and one for nucleic acids. In each room, you must list 2 foods that you can find that macromolecule in, the elements that make up that macromolecule. You must draw, don't print out a picture and paste it. You must draw a monomer. Okay, so that would be the monosaccharide for the carbohydrates. You must draw either triglyceride or phospholipid or steroid for the lipids. You must draw the nucleotide. You must draw an amino acid. Okay, you must draw it. Do not print out a picture. Then you're going to give the components of the monomer if it has components, like the amino acid and the nucleotide both have 3 parts that make them up, right? So for the amino acid, you need to list that it has an amino group and a carboxyl group and an R group that varies. For the nucleotide, you need to list that it has a 5-carbon sugar, a nitrogen base, and a phosphate. So you want to make sure you list those things for the monomers that have parts to them. That's only the monomer for protein, the monomer for nucleic acids are the only ones that have parts that build them up as well. Then you have to tell specifics about the structure and function of the ones that's listed on the page. So I want a specific for the carbs. I want specifics about cellulose versus starch. For lipids, I want you to do saturated versus unsaturated fats. For proteins, you're going to tell how the R groups on the different amino acids interact. You can use your notes. You can use your online textbook. You can Google. That's fine because you're learning while you're looking up this information. What you can't do is copy and paste it on a document and glue it on. Everything must be done by hand. You learn more when you actually write, okay? For nucleic acids, you're going to talk about DNA versus RNA. After you do that, you're going to give 2 examples of polymers. Now, for nucleic acids, you're going to have to do DNA and RNA because I haven't talked to you about any other examples of nucleic acids. Then you're going to give the function of the polymers that you actually listed. Yes?
For polymers, for lipids, what are we going to do?
For polymers for lipids, you're going to give examples. So you may say a specific saturated fat for a lipid. For the phospholipid, you have to say the cell membrane as an example. For steroids, you might want to list cholesterol or testosterone or estrogen. But for the lipids, you're going to give an example. That's a good question because they don't really have polymers because they're not true polymers. Your rooms must be full. So that space needs to be full. It must be colorful. And it must have at least 2 interactive components. So that means if you want to do a flip, like, it's closed and I have to open it up to see what's on the inside, or if it's going to have a little pull out. Okay, so you can do the fold outs. You can do a pocket where I got to open the pocket and pull a card out. But you got to have at least 2 interactive parts. You can't just draw stuff on the poster and leave it there. Y'all understand? It needs to be full. You see examples on this page. I know it's in black and white, but if you click on the notebook in Canvas, you can see it in color. It's easier to see when it's in color to kind of see an example of what one looks like. Yes?
On the kid, carbohydrates, take 2 places.
No, that's one room. That's the whole room. It ends up with, you got 2 walls in the room. Each one of your walls will have each one of your rooms will have 2 walls that you need to fill up. And the floor. Y'all understand? When you fold it, each room, you'll have it's like putting this through the middle, but each one will have 2 walls and a floor, and it needs to be full. Okay? One thing you may want to do, like what somebody did, was they got the little plastic foods, come with a play kitchen, and they actually glued those in on the different foods for each one of the, so be creative, guys. This is a major grade. It's a test grade. Okay? I'm posting the rubric in Canvas today. You know how at the top of the page where it gives you a link to this page? The rubric will be on that page. So it'll be at the top of every page, a link to the rubric. So you'll know exactly what you need. Yes?
And all of that needs 1 poster.
That's 1 poster.
Just 1 poster.
That's 1 poster. That's all you need is 1 poster. You cut it, you fold it, then you make 1 slit, and then the way you fold it, it pops up like that. That's 1 poster. All you need is 1. All right. Huh?
I said it looks like it needs a lot more.
Nope, it's just 1. It's all in the folding. All right, now, let's review. I'm going to tell you the same thing I told my other classes. You need to be able to do this without looking at your notes. And that's how you'll know that you know your macromolecules, okay? Today, at the end of class, there will be a chart in Canvas that you can print out. If we have time, we probably will in this class. I'll print out some for you as well. And then you just copy that chart over. It'll be a blank chart, nothing on it. But you copy it over and over and over again. You start off using your notes. You're copying it from 1 page to the next page. And then you see if you can do carbs without looking. Once you get carbs down without looking, see if you can do lipids without looking. If you can do that, then you're actually learning the information, and the act of writing it is making muscle memory, which gives your brain another connection to the information. Okay? That will help you learn this information. If you need a scratch paper or a blank chart on test, like if you've done that and you want me to give you a blank chart so you can sit there and write it out before the test and then use that, that's fine. Because that's not cheating. If you sit there and write it out from your brain, you can't use your notes to write it out. Let me be clear on that. But if you're getting ready to start the test, you're like, Ms. Young, I need my chart, I'll give you a blank one. And you can write whatever you want to on there and then use that on your test. This is a good way to make sure you learn this information. Are y'all with me? Let's go over it real quick before we start talking about enzymes. All right. So there's 4 major organic compounds. We have carbs, lipids, proteins, and nucleic acids. What are the elements that make up a carbohydrate?
Carbon, oxygen, hydrogen.
That's it. All right. What's the monomer called? Monosaccharide. Give me an example other than glucose. Can y'all give me one other than glucose? Cellulose is a polymer. But ribose, deoxyribose, fructose, galactose, those are all examples. I knew y'all were going to say glucose because it's up there. All right. What's the polymer? Polysaccharide. Give me an example other than starch and cellulose. What is it? Chitin. And what's the other one? Glycogen. Good job. All right. Those are the 4 we talked about. We talked about starch, cellulose, which are both found in plants, glycogen and chitin, which are both found in animals, and chitin is also in fungus. All right. What's the function of a carb? Short-term energy storage. Okay. Now, I know it says monomer and polymer up there, but we know that lipids are not true polymers because they don't have monomers, but we're just going to fill in the chart. There's 3 classes of lipids that we talked about in detail. We mentioned waxes, but waxes are used for protective covering. But in detail, we talked about the triglyceride, the phospholipid, and the steroid. T is for triglyceride, P is for phospholipid, S is for steroid. Y'all understand that up there? All right. Now, what are the elements found in lipids? That's it. Carbon, hydrogen, oxygen, and sometimes phosphorus, which is why it's in the parentheses. Which one of those 3 up there has the phosphorus in it? The phospholipid. Cool beans. All right. What are the components that make up the triglyceride?
Carbon.
Not the elements, but the components. Glycerol and what? 3 fatty acids. Glycerol and 3 fatty acids make up a triglyceride. What's the common name for triglyceride? Fats and oils. I love it. Y'all making me so proud. All right. What are the components of the phospholipid?
Carbon.
Not the elements, but the components.
2 fatty acids.
2 fatty acids, glycerol and phosphate. That's it. All right. How do I identify a steroid? What will I see? Not the nitrogen base. There's no nitrogen in lipids, okay? But you're talking about the 4 fused hydrocarbon rings, okay? When you see those 4 fused rings together, that lets you know that's a steroid, okay? All right, now let's talk about examples. What is an example of a triglyceride? Saturated and unsaturated fat. How can you tell the difference? Which one do plants have, saturated or unsaturated? Unsaturated. And what do they look like at room temperature? Liquid, that's right. Which one do animals have? Saturated. And what do they look like at room temp? Solid. Cool beans. What's an example of a phospholipid? The cell membrane. What's an example of a steroid? Cholesterol. And there's also the sex hormones, which would be estrogen, progesterone, and testosterone, okay? So sex hormones are also examples of steroids. All right, what's the function of a triglyceride? Long-term energy storage. And what are the other functions for triglycerides? Insulation, and what else? Protection of those internal organs. Good job. So the main function is long-term energy storage, and then you also have insulation and protection of your internal organs, okay? All right, what's the function of a phospholipid?
Creates cell membrane.
Creates a cell membrane. Cool beans. And what is the function of steroids?
Cell communication.
Cell communication, chemical signaling. Good job. All right, let's move on to proteins. What are the elements that make up a protein?
Carbon, hydrogen, oxygen, and nitrogen.
Carbon, hydrogen, oxygen, and nitrogen. All right, what's the monomer for a protein? Amino acids. Proteins are amino acids. Amino acids have 3 important parts to them. What are they?
The amino group.
The amino group.
The phosphorus group.
Not the phosphorus group, but the
Chunks.
carboxyl group.
Yeah, start with the C.
And what's the other one?
The R group.
The side chain, which is the R group. All right, y'all killing it, for real. What's the polymer called? Polypeptide. All right, and an important polymer example is what we're talking about today, which would be enzymes, okay? Enzymes are special proteins. We would not be able to live without them. So that's an important example, and we're going to talk about enzymes specifically today. All right, proteins have about 8 different functions, but what are the 3 I told you I needed you to understand? Structure, catalyst, and immunity. Now, the enzyme's job is to function as a catalyst, okay? So the enzyme is directly connected to the catalyst. That enzyme is a catalyst. It's a catalytic protein. That's what enzymes are, a catalytic protein. All right, last one, the nucleic acid. What are the elements of a nucleic acid?
All 5 of them.
Yeah, all 5 of them. Carbon, hydrogen, oxygen, nitrogen, and phosphorus. Which 3 elements are found in all 4 major organic compounds? Carbon, hydrogen, and oxygen. What's the monomer for a nucleic acid? Nucleotide. Nucleotides have three parts that make them up. What are they? Nitrogen base, phosphate, and 5-carbon sugar. Cool beans. All right. What's the polymer called? Polynucleotide. And what are the examples?
DNA and RNA.
DNA and RNA. All right. What's the function of DNA?
To create protein. No, that's RNA. That's cell communication and.
Not cell communication. Store and transmit genetic information. And you already said RNA, which is to help do what? Create proteins. Cool beans, guys. You guys are awesome. You really are. All right. Got a question?
For the proteins, you said the examples are enzyme and what?
Only gave one example for enzymes, but proteins could be stuff like the keratin in your hair, your muscle. There's so many different proteins because they all have different functions. Like you could have the membranes the proteins that's embedded in the membranes for transport. But I specifically told you enzymes because we're going to actually talk about enzymes in a few seconds.
I think it's refusing.
All right. Did I say what?
I think it's refusing.
Oh, and I just wanted to make sure I wanted to overrate that. All right, so let's talk about enzymes, okay? Enzymes are a special class of what we call catalytic proteins, okay? They're a special class of what we call catalytic proteins. So since they're proteins, what are they made of?
Cells.
Amino acids, right? They're made of amino acids because proteins are made of amino acids and an enzyme is just a special type of protein, so they're made of amino acids. Now, let me give you a scenario. So, I need 3 cases of paper from Walmart. This is not true, I'm just saying I do. Well, I actually need 6 cases. I'm going to send Paige and I'm going to send Araya to Walmart for me. Araya is going to walk, but Paige is going to drive my car. Okay? They're both going to Walmart. They're going to pick up 3 cases of paper each. Okay? They're leaving from the same exact spot. They're leaving from room 216. They're going to Walmart. They're both going to get 3 cases of paper. Araya is going to walk. Paige is going to drive my car. Who's going to get there first? Paige. Who's going to use less energy to get there? Paige. What's the difference? They're doing the exact same thing, but what was the difference? One of them is using a car. The car in this scenario, so we got, what y'all laugh? That's Paige.
You gotta drop me real big.
Girl, this is Araya. They're both going to Walmart for 3 cases.
About it, walk side me, no.
Leave me alone. Alright. Paige is going to drive the car.
Not bad.
Leave my car alone. Y'all, Paige is driving the car and Araya is walking. The difference between, so you said that Paige is going to get there faster and she's going to use less energy. And the difference maker was the presence of the car. The car in this scenario represents an enzyme. Okay? That's what enzymes do for chemical reactions. So what I'm doing here, the starting point is called my substrate. This is my substrate. The substrate is whatever you start with, okay? The substrate is what you start with. My ending point is called my product. The product is whatever you end with. Now, is my starting point the same?
Yes.
So my substrates are the same. Is my product the same?
Yes.
So my products are the same, but the top reaction happens faster using less energy because there is an enzyme present. That's what a catalyst is. A catalyst is something that makes a chemical reaction happen faster by requiring less energy to get it started. Okay?
Can it be a negative thing?
Not an enzyme. All right. So, every chemical reaction requires some energy to get going. That's called the activation energy. Okay? It absorbs energy from the surroundings. An enzyme lowers the amount of energy that's needed to get a reaction going. If I need less energy to get going, that means I get started faster, so the reaction itself is actually faster. That's the difference between you eating a sandwich and it digesting in your stomach with digestive enzymes, or sitting a sandwich on a table and watching it break down. It is going to break down regardless, but which one's going to happen faster? In your stomach, because you have digestive enzymes that help that reaction happen faster, okay? So that's what enzymes do. Enzymes are catalysts, and catalysts help reactions to happen faster by requiring less energy. So we have a little, y'all see how great my artistic work is already, right? Yeah, because I am the best. All right, so, two reactions. All right, now, this represents from this line up, from here up, so either to here, from there to there, or from here to here, represents the amount of energy needed to get this reaction going. So this is my energy of activation, my EA. Which one of these needed the less amount of energy, the green or the yellow?
The green.
The green one. So, which one of these represents, my bad, y'all. Take that out. All right. Which one of these represents the enzyme catalyzed reaction, the green or the yellow?
The green.
The green one. So, since it got started faster, it also reached the end point faster because it needed less energy. Okay? Do y'all understand?
It's like a boost.
It's like a boost, exactly. It's like a boost. It's a boost to get it going faster. Okay? Now, enzymes themselves are specific for several things. They're very, very picky. Okay? So I'm going to draw me an enzyme on the board. Remember, this is a catalytic protein. So this represents my enzyme. Now, what's a substrate? It's the thing you do what? Start with. So I have 3 substrates up here on the board. There's 3 substrates. So an enzyme have what's known as an active site, okay? The active site on the enzyme is the location where the substrate actually binds to the enzyme. It's where the substrate makes a bond to the enzyme. It's specific, okay? It's called actually a lock and key. So which one of my 3 substrates, A, B, or C, will actually fit the active site? A. You said definitely C. Yeah, I thought it was C too, girl. I tried it. It didn't fit. All right, so definitely A will fit the active site. Now, when the substrate actually bonds to the enzyme, so we know that A is the one that fits in there. Now I call this an enzyme substrate complex because it's actually the enzyme and the substrate together. when the enzyme and substrate bind, it's called an enzyme substrate complex. The conditions in this enzyme make it easier for the substrate to do what it's supposed to do. In this case, I'm going to do a digestion. What does digestion mean? Build up or break down? Break down. Is that a hydrolysis or a dehydration? Hydrolysis. So what's going to have to come in to help this out? Water has to come in and help it out, because since I'm breaking down, this is a hydrolysis reaction. Now, when this is released from the enzyme, it'll no longer be the same thing that went in. Now it is my product. Okay? Y'all with me? Now, the enzyme, though, does not change during the reaction. The enzyme stays the same so that a new substrate can bind to the active site. Are y'all with me? It does it over and over and over again as long as there is substrate that needs to be catalyzed. Okay? Now, what's the thing that we start with called? Substrate. What's the thing we end with called? Products. All right. What's not changed during the reaction?
Enzymes.
Enzymes are not changed. Enzymes are proteins, and since they're proteins, they're sensitive to their environment. Okay? Now, if you change the environment of a protein too much, it can cause a protein to do what's called denature. And denature simply means to unfold. The problem with that is structure dictates function. Say that. Structure dictates function. So what happens if I change the structure of a protein by unfolding it? You change the function. Okay. So there are three things that can make a protein or an enzyme denature. If I change the pH by too much, so that's how acidic or basic it is. Think about it. Enzymes in your stomach work in acid, but enzymes in your blood work in a neutral environment. So if you put those enzymes in the wrong location, they don't work because they begin to denature. Okay? The second thing that can cause a change is temperature. This is why a high fever or a low fever for a long amount of time is dangerous, because it causes your proteins in your body to start to unfold. That can lead to seizures and things like that. It can actually lead to death if the fever is too high for too long. Low grade fever, not the same thing as a high fever. I'm talking about like a high fever.
Like 300?
If your fever 300, you've been dead. You've been dead a long time. Matter of fact, they probably cremating you. That's not a fever. You being laid to rest. All right. The other thing is salinity. Do you know what salinity means? Salinity is the salt. Good job. The salt concentration. All right. The amount of salt. Okay. Those three things can cause an enzyme to denature. Enzymes work within a range. So if I have an enzyme, and let's say this is pH, so I'm going to do 1, 2, 3, 4, 5. So this is my pH, and this is my enzyme activity. So let's say the enzyme, I'll put two of them up here.
So salt is not good for you?
It's in certain amounts. I mean, you can get, there's a range, you need some sodium and chloride ions in your body for your body to function properly. But too much is not a good thing.
Like one bottle?
Definitely don't eat a bottle of salt. Please don't. All right. So I have two enzymes here, okay? I have enzyme A and enzyme B. Well, reaction A and reaction B catalyzed by enzyme A and enzyme B. So this is my enzyme activity. Now, enzyme A works between a range of 1 and about what? About 1 and about 3. Okay, y'all see that? So this is its range. It will work the entire range, but it works best at about what? 2. This is called optimum. This is the optimum condition for that enzyme. The place where the activity is the greatest is the optimum conditions, okay? This is where the enzyme works best. It'll work at 1. It'll work almost up to 3, but it works best at 2. Once you get outside of this range, it'll begin to unfold. What's the range that both enzymes A and B will work in? 2 to what? 2 to 3. They'll both work, not well, but they'll both work in between a range of 2 to 3. Okay? Y'all see that? All right. So these are the 3 things that they're specific for that can make it denatured. There's 2 more things that enzymes are specific for as well.
So like
Alright.
it's like it's running in the beginning, it's running slow, then it's heating up at the top, then it's becoming slow.
And then it'll start to slow back down once you start getting past that optimum level. Yes. I don't know why I just didn't do that. All right. Now these next 2 things, enzymes are also specific for. They're specific for the shape of the substrate. That's called what? We talked about it earlier, lock and key, remember?
Yeah.
It's specific for the shape of the substrate, and it's also specific for the concentration. All right, and what I mean by concentration, that's the amount of substrate versus the amount of enzyme. Okay? If a reaction starts to slow down, you put in more substrate, it'll speed back up. Okay? But if you take out substrate, what's going to happen to the reaction? It's going to slow down. Okay? So there needs to be a good ratio of enzyme to substrate to make sure that the reaction continues to happen at the correct pace. Y'all with me?
Yes, ma'am.
Cool beans. Turn to page 90. We're on page 90. Practice 5. Practice 6 is your homework. Practice 6, finishing up your study guide. You should be able to finish those questions and working on your project. That's your homework. Okay? If you want to do the extra credit, that's going to be in Khan Academy. I'll push the activities out when I go back to my desk. All right. We're going to do this together as a class, and we're going to say the actual thing, not the letters. We're not going to say foxtrot or alpha or anything like that. We're going to say, like, if it's active site, we're going to say active site, okay? We're going to go across the top, and then we'll go across the bottom, and then I'll go back and make sure that everybody has the right answers because I want to make sure you're studying the right thing.
Yes, ma'am.
Cambo, you good?
Mhm.
All right. All right. Let's start at the upper left-hand corner. That's pointing to that whole molecule right there. What is that?
The exercise.
That's the substrate. That's what you start off with. That's the substrate. All right, so you can write substrate or you can write foxtrot there. All right, that is the substrate. All right, let's move on to the next one. That's pointing to that yellow, to the whole, y'all's not in color, I forget. Yeah, that's pointing to that whole thing. What is that? Enzyme substrate complex. That's them together, okay? That's the enzyme and the substrate together. All right, the next one is pointing to what's that going in? Water. So what process is that? Hydrolysis. Good job. That one is hydrolysis. So is this going to be breaking down or building up? Breaking down. If water was coming out, it would be dehydration, and you would be doing what? Building up. Good job. All right. Now we see the things that came out of the enzyme substrate complex, and what are those? Products. All right. Let's go down to the bottom. It's pointing to that space that's inside of that thing. What is that space called? The active site. And what is that thing? That's the last one. What is that thing? An enzyme. An enzyme is a special type of what? It's a protein, so it's made up of amino acids. Good job. All right, y'all. Say the answers. I'm going to go through it. The answers across the top. Foxtrot, Charlie, Delta, Echo. That's across the top. The 4 lines across the top. Foxtrot, Charlie, Delta, Echo. The two lines at the bottom, Alpha, Bravo. Okay? Now, listen up. Thought y'all laugh at me. Alright? Got a science song for you. Don't laugh at me. Some stuff that's in my body's breaking down no matter what. Gotta change a substrate into a product. And every time an enzyme's in the mixture, rate reactions will go up. Energy goes down, energy goes down, energy goes down, up, down, up, down, up, down. We mix the enzymes in. The substrate's used up, changed into a product. Enzymes still there. All right, y'all finna sing it with me. One time, y'all gonna do it with me. Okay, the first line says, some stuff that's in my body's breaking down no matter what. Y'all got that?
Yes, ma'am.
You got it, Cam. 1, 2, ready, go. Some stuff that's in my body's breaking down no matter what. Second line, gotta change a substrate into a product. Ready? Go. Gotta change a substrate into a product. And every time an enzyme's in the mixture. Did y'all hear that?
Yes.
1, 2, ready, go. And every time an enzyme's in the mixture, rate reactions will go up. 1, 2, ready, go. Rate reactions will go up. Energy goes down. Energy goes down. Energy goes down, up, down, up, down, up, down. Y'all ready? 1, 2, ready, go. Energy goes down. Energy goes down. Energy goes down, up, down, up, down, up, down. Last part says, we mix the enzymes in. 1, 2, ready, go. We mix the enzymes in. The substrates used up, changed into a product. Enzymes still there. 1, 2, ready, go. The substrates used up, changed into a product. Enzymes still there. So what that song tells you, and again, this is on my webpage, Young Science Rocks, and I'll put the words in Canvas if you want to use it. You're welcome to look at it. I promise you these songs help. It's telling you that the reaction is happening, changing the substrate into a product, but when an enzyme is in the mix, the rate of the reaction goes up and the energy goes down. So the substrate will be used up because all of the substrate is changed into a product, but the enzyme is still there because enzymes remain unchanged in a chemical reaction. Okay? Now, one last thing before I go to my seat. So, we talked about the digestive enzymes specifically, okay? Most digestive enzymes are going to end with ACE, okay? So, if I have lactose, which is milk sugar, the enzyme that is going to break it down is called lactase. If I have sucrose,
Then I'm gonna have sucrose.
which is table sugar, the enzyme is going to be sucrase. So what if I'm trying to break down malt sugar or maltose? What's the name of the enzyme? Maltase, okay? Sugars, most sugars end with ose. So those are carbohydrates. Most digestive enzymes end with ACE, and those are proteins. Don't confuse the two. Ose is carb, ACE is protein, okay? When I see ose, I think carb. When I see ACE, I think protein, specifically enzymatic proteins. Ose equals carb, ACE equals protein. Y'all with me? All right, cool beans, guys. Don't forget, practice 6 is your homework, and to finish the study guide, and also work on your projects. Thank you for watching this to make sure it didn't fall.